Webinar today. My name is Nancy Carrico, and we'll be talking about the Nextera XT sample preparation today. So by the end of this webinar, you should be familiar with the Nextera XT protocol and the best practices for a successful library preparation. So just to go over what we're going to be talking about today, we're going to go over a brief introduction about the Nextera XT DNA sample preparation kit. Then we'll go over some comparisons between the Nextera DNA and the Nextera XT DNA library preparation kit. We'll go over what components are necessary to complete the protocol. And then we'll go over each step of the protocol and then the best practices that are required for each step. We'll discuss some alternative normalization methods, and then at the very end, we'll provide you with some additional resources that you can always go back to. All right, so just to start with a brief introduction, Nextera XT DNA Library Preparation Kit is one of our most popular kits that we provide, and it is a very versatile library preparation kit. So it can be used for preparing libraries from genomic DNA of small genome organisms, plasmids, CDNAs, or amplicaons. It's a very fast workflow, so it can be completed in less than 90 minutes. Also, of all of the library preparation kits that we have available, it starts with the least amount of starting material, so just 1 nanogram. And because we start with such a small quantity of DNA, we do ask you to use a fluorescence-based method for quantification. We have the capability of multiplexing up to 384 samples with this kit using our dual indexing strategy. Also, another plus side is that it uses an enzymatic fragmentation mechanism, so we don't need any mechanical shearing equipment such as a sonicator or Covaris that can be quite pricey. It's very high throughput friendly as well and can be optimized for automation. And the reagents are master mix, so it minimizes pipetting steps, hands-on time, and again allows for a very fast workflow. So we have 2 different Nextera kits that have very similar names. So I just wanted to point out the differences and when you would want to use one over another. So the Nextera DNA library preparation kit starts with 50 nanograms of input DNA, and the Nextera XT DNA library preparation kit starts with 1 nanogram of input DNA. They have very similar workflows, but they were optimized for the different starting material amounts. So we recommend to use the different kits for different applications. So for the Nextera DNA library preparation kit, we would recommend to use with large or complex genomes, such as those listed here, such as human or non-human mammalian genomes, plant or invertebrates. For the Nextera XT library preparation kit, it can be used for small genomes, amplicaons, or plasmids. When we're using PCR amplicaons as the input material, we do ask you to use an amplicaon that's at least 300 base pairs in size. And the reason for this is because there is an expected drop in sequencing at the very distal 50 base pairs or so from each end. The reason for this is because the transposomes cannot add the tagging sequences at the very end of your amplicaon. And so the way to get around that would be to design your PCR amplicaon to have an extra 50 base pairs on each end, and then we can avoid this drop in coverage. The Nextera XT kit can also be used for plasmid DNAs. It is compatible with circular double-stranded DNA, even for very large circular double-stranded DNA such as BACs or bacterial artificial chromosomes. The kit can also be used for microbial genomes, such as bacteria or viruses, and it's also frequently used for library preparation from double-stranded CDNA as well. So to complete the Nextera XT DNA sample preparation, you will need a couple different kits. First of all, you will need the Nextera XT DNA sample preparation kit, we have two different configurations, a 24 sample kit and a 96 sample kit. So these kits will contain the enzymes and all the core reagents that are necessary for library preparation. In addition to the Nextera XT DNA sample preparation kit, you will also need the Nextera XT index kit. So even if you're not planning to index multiple samples or pool a lot of different samples, you still need these index kits to be able to complete the library preparation, and you can use any of the 6 kits that I have listed here. So we have version 1 index kits as well as version 2 index kits. These kits will work exactly the same, but the only difference is that the Nextera XT V2 index kits, they came out later to allow for higher multiplexing. So if we pull all the indices from set A, B, C, and D, we can get up to 384 plex pooling. So one thing I do want to point out here is that the 96 index kit, it doesn't mean that it's coming with 96 individual tubes of indices. It comes with 12 I7 indexed oligos and 8 I5 indexed oligos. And so the combination of the 12 and 8 I7 and I5 indexed oligos will be able to give us the 96 index combinations. Similarly, the 24 index kit comes with 6 I7 indexed oligos and 4 I5 indexed oligos, giving us 24 index combinations. And I just want to point out again, you will need both the I7 and I5 index oligos to complete the library preparation. In addition to the kits, we may also want to have a TruSeq index plate fixture kit, as you can see here in the illustration. So this will hold your I7 and I5 index tubes, as well as your PCR plate, and it'll make your life just a little bit easier where you can use a multi-channel pipette to pipette in your indices into the PCR plate. And depending on the platform or sequencing platform that you're hoping to sequence your libraries on, you may also need to purchase sequencing primers. So I just want to make it really clear here that you will only need to purchase these sequencing primers if you're planning to use a HiSeq 2500 or lower with the TruSeq V3 high output chemistry, okay? So if you're using any other chemistry version or any other platform, you do not need to purchase any additional sequencing primers. They are already included in the reagent kits. But in the case that you are using the HiSeq 2500 with TruSeq V3 high output, you can order either the single read or paired end read sequencing primer kits here. So once you order the kit, you will receive 3 different boxes. And I just wanted to point out here that there are some reagents that are shipped and stored at different temperatures. So box 1 will ship on dry ice, frozen. But the LNW1 included in this box, we recommend to store it at 2 to 8 degrees. So we would want to move this tube into the refrigerator and store it at the proper temperature. Similarly, box 2 will ship at room temperature, but the LNB1 in this box, we recommend to store it at 2 to 8 degrees. Again, make sure it's stored at the correct temperature for proper usage. One mistake to avoid is to not mistakenly put it in the freezer, because the LNB1, once frozen, will not be usable. The index kit will ship on dry ice frozen, and you can just put the whole box into the freezer. All right, so to briefly go over the workflow, we're going to start with the input material. This will then go through a Tagmentation reaction. So Tagmentation stands for tag and fragment. So the DNA will get fragmented and tagged. This Tagmented DNA will then go through a PCR amplification with our Nextera XT index PCR primers. These amplicaons will then go through a cleanup step using AMPure XP beads to remove any small or unwanted fragments. And then these cleaned up libraries will go through normalization and then pooled and proceed to sequencing on our different platforms. It is a fairly short workflow, but you may still want to stop in the middle of the library preparation if you have a meeting, you need a coffee break or whatnot. So these are the steps that we would recommend to stop if you must. So after the amplified tagmentation step, the samples will be on the thermocycler, so you can leave it there overnight, or alternatively, put it at 2 to 8 degrees for up to 2 days. Otherwise, you can store the cleaned up amplified DNA or the normalized DNA at -25 to -15 degrees for up to 7 days. And these might be good places to stop if you need to send your sample to a facility for sequencing, for example. All right, so to give you a little bit better understanding of how the library preparation works, we're going to be mixing our genomic DNA with transposomes. And the transposomes will sit on our double-stranded DNA fragment at random locations, introduce a double-stranded DNA break, and then ligate on our tag sequences. These tag sequences are actually partial adapter sequences that will be recognized by our Nextera XT index PCR primers, as you can see towards the bottom. And then once it goes through the amplification step, it will complete the adapter sequence, as you can see at the very bottom illustration. So the P5 and P7 sequences you see here are important for recognizing the oligos on our flow cells and clustering. The index 1 and index 2 sequences are important for demultiplexing our different samples that are pooled together. And then the read 1 and read 2 sequencing primer binding sites, these are where the sequencing primers will bind and read our insert as well as index sequences. So for our sample input, we require to use at least exactly 1 nanogram of high-quality genomic DNA. And in the case we're using amplicons, we ask you to use amplicons that are at least 300 base pairs or longer, because shorter fragments may get over-tagmented and lost during the subsequent wash steps. So some best practices for preparing your sample input. Because we start with such a small quantity of starting material, we do ask you to use a double-stranded DNA-specific fluorescent dye. So you can use equipment such as Cupid or Picogreen to measure your starting material input. And we also recommend to use molecular grade water or 10 millimolar tris at pH 7.5 to 8.5 for optimal tagmentation reactions that are following this step. A well-known inhibitor of the tagmentation reaction is EDTA, and EDTA is commonly present in a lot of elution buffers that come in DNA extraction kits. So TE and AE, for example, are some of the buffers that you may be familiar with. And you want to avoid using any of these buffers that may contain EDTA because they will inhibit the enzymatic activity. There are a couple of different other chemicals that may inhibit the tagmentation reaction, such as phenol or ethanol, high salt concentrations, polysaccharides, depending on the sample that we're starting with. So we want to make sure we're starting with a pure DNA sample, and the purity can be assessed through measuring the absorbance ratios at A260 over 280 and A260 over 230, and we want the values to be within the ranges indicated here. And it's also important to make sure our pipettes are accurately calibrated and we can trust the volumes that we're pipetting and mixing. So the next step is the tagmentation reaction. We'll be mixing our double-stranded DNA starting material with buffer and transposome in a 20 microliter reaction. These will incubate at 55 degrees for 5 minutes. And what's happening during these 5 minutes is our transposomes will be sitting on our genomic DNA at random locations, introducing a double-strand cut, and then ligating on those partial adapter sequences or tags. Some best practices for the Tagmentation reaction. We want to make sure our thermocycler is calibrated because the Tagmentation step is sensitive to temperature. And there is a 10 degrees hold after the 5 minutes of incubation. Our transposome is still active at this temperature, so this is not a good step to take a break or leave the sample on the thermocycler for a prolonged period of time. We want to immediately proceed to the neutralization step. And so for the neutralization, we'll be adding the NT buffer and incubating at room temperature for 5 minutes. After the Tagmentation, we're going to do the PCR amplification. This step will require the 2 index PCR primers, and they will go through a 12-cycle PCR reaction. And again, we're just going to stress here, we need both primers, the P5 or the I5 and I7 index primers to complete the PCR amplification step, as well as complete the adapter sequences. So you can see here towards the bottom, during the Tagmentation reaction, we're going to be adding on the read 1 and read 2 sequencing primer binding regions. But during this PCR step is when we add on the P5, P7, as well as the index sequences. And so we want to make sure we're using both primers to complete the adapters and complete the library preparation. Some best practices for the PCR amplification step. Make sure to use a heated lid. If we don't use the heated lid, we may see evaporation and reduced yields. And then also make sure your thermocycler is accurately calibrated. Okay, so after the amplification, we're going to clean up our sample using AMPure XT beads. So the goal of this step is to make sure we're removing any small fragments or primary dimers, unwanted fragments from our libraries that may affect our sequencing quality. And you can find this table in your reference guide as well, but depending on the different input sizes, and by input size we mean the size of the starting material that we started the library preparation with. And so for example, if we're using genomic DNA as our starting input, we would recommend to use 30 microliters of AMPure XT beads to the 50 microliters of your sample at this point. So that would be a 0.6x AMPure XP ratio to get an optimal cleanup. And for example, if you started with a 400 base pair amplicon sample, then you would want to be using 90 microliters of AMPure XP beads to the 50 microliters of your sample at a 1.8x ratio. Following best practices during this AMPure cleanup is very important to make sure we're cleaning out any unwanted fragments as well as not losing any of our important libraries that we want to be sequencing. So some best practices are to make sure your beads are at room temperature before we use them. So they should be stored at 2 to 8 degrees at a cold temperature, but bring them up to room temperature before we use them. Also make sure our beads are homogeneously resuspended, so vortex vigorously before we use the AMPure XT beads. We want to make sure we're not carrying over beads to our final library. So pipette slowly, make sure we're not removing any beads off of the magnet when we're taking off the supernatant from the beads. Also, it's important to use a strong enough magnet to make sure that we're not losing any of our library. So we have some recommended magnets in the reference guide. We would recommend to use those magnets to make sure that we get good cleanup. And it's also important to make sure we're freshly preparing our 80% ethanol before cleanup. So once we've cleaned our libraries, at this point, our libraries are double-stranded DNA, and we can quantify the libraries using a fluorescence-based method such as qubit or picogreen. And also we can take a look at what our library looks like by using a bioanalyzer or similar equipment. This is what some example libraries would look like after we've finished the library preparation at this point. And so you can see that the traces look quite different. The top one has much of a broader library trace, whereas the one on the bottom has sort of a peak towards the 1,000 kb size range. So these are all typical library traces that we would expect from a Nextera XT library preparation, and you can find these example traces in our reference guide as well. So as long as we have a library trace that's within 200 base pairs to 1,500 base pairs that can cluster on our various different platforms, they are perfectly good to proceed to sequencing. But if you have any doubts, you can always contact us at tech support, and we'd be happy to take a look and make suggestions. If you are interested in knowing what the insert size may be, the adapter length all added together is about 135 base pairs. So the insert size would be your library size minus the 135 base pairs of adapters. Because we use enzymatic fragmentation, the amount of starting material that we start with can affect the final library trace. And so if we started with the recommended 1 nanogram of starting material, again, this is what we would expect to see, a library trace that starts at 200 to about 1,500 base pairs in size. But if we started with too little sample input, so less than 1 nanogram of starting material, we may see that the final library trace has shifted towards the left or smaller fragment sizes, and this is what we would consider an over-tagmented library. On the other end of the spectrum, if we started with too much starting material, so greater than 1 nanogram of starting input, we may see that the library trace has shifted towards the right or larger fragment sizes, and this is what we would consider an under-tagmented library. So these under-tagmented libraries can be a little more tricky to get good sequencing from because our platforms don't cluster large fragments very efficiently, so they may require a little bit more optimization. So in order to get a good library profile within our expected range of 200 to 1,500 base pairs, we have a document here that I linked, the Nextera XT Library Prep Tips and Troubleshooting Guide. There's a lot of great information here on how to get an optimal fragment size range using the Nextera XT Library Prep Kit, so I would highly recommend taking a look at this Tips and Troubleshooting Guide. So once we've taken a look at what our library looks like, we can proceed to normalization. And so the goal of normalization is to have all our libraries at equimolar concentrations when we're pooling and denaturing our samples to sequence on our different platforms. And there are two different normalization methods that you can use for the Nextera XT library preparation protocol. There's bead-based normalization, and then there's manual normalization. So if you're choosing bead-based normalization, you would follow the Nextera XT reference guide, the normalized library section, and then proceed to protocol B in each system's denature and dilute libraries guide. If you're using manual normalization, and we'll discuss how you would manually normalize your libraries, but once you've done that, you can proceed to protocol A in each system's denature and dilute libraries guide. So when would you want to use bead-based normalization? So the advantages of bead-based normalization is that it's very useful for high throughput preps. If we have a lot of samples, it doesn't require you to manually quantify or visualize each different library. So it's going to save you a lot of pipetting and a lot of time. However, some disadvantages is that once we have performed the bead-based normalization, the libraries that are eluted off of the normalization beads are single-stranded DNA. So once we've finished the bead normalization, the single-stranded DNA cannot be visualized on a bioanalyzer, and they can't be quantified using double-stranded dyes. So it may be difficult to get good QC once the bead normalization has been performed. Also, because it is single-stranded DNA, it's not as stable as double-stranded DNA, and it will not store as long, and it will be more sensitive to freeze thaws. So if you are planning to maybe store these libraries for a long time or send it off to a sequencing core that might require you to freeze thaw multiple times for whatever reason, this bead-based normalization may not be the method you would want to use. So how does bead-based normalization work? And so this is a little animation that might help you understand a little bit better how this bead normalization process works. And so sample A and sample B, you can see in the Eppendorf tubes down there that the library concentrations are a little different. So sample A, we have some more libraries compared to sample B. So to the two samples, we're going to add our normalization beads. And to the beads that have been added, only a certain amount of library can bind. Any library that didn't bind to the normalization beads will get washed away. And then the libraries that are bound to the beads will then get eluted off, and we'll have equal amounts of library from each sample going into the pooling step. So some best practices for bead-based normalization. It is critical to make sure that we're adding the same amount of LMB1 to each tube. So we want to make sure LMB1 is completely resuspended and homogeneously resuspended before we're adding them to the tubes. So make sure we're mixing it completely before using them. LMB1 can be a little viscous, so we don't want to be using P200 pipette tips. They may get stuck in your pipette tip. We may not be accurately pipetting accurate amounts of the LMB1 beads. So we would recommend to use wide bore pre-cut tips or a P1000 pipette. It's also important that our microplate shaker will go up to high speeds. If we can't get up to 1800 RPMs, we may not get good mixing. The libraries may not bind very efficiently or elute efficiently from the LMB1 beads. So make sure your microplate shaker will go up to 1800 RPMs. And then the normalization is not very efficient if your library is less than 15 nanomolar concentration. And so because it is a site saturation binding reaction, if we don't have at least 15 nanomolar to saturate the binding sites of the beads that are being added, we may not get even representation after the sequencing has been performed. So we want to make sure we're starting with at least 15 nanomolar concentration. And again, as I mentioned earlier, once the libraries are eluted off of the beads, they will be in single-stranded form. They are not as stable as double-stranded DNA, and they're more sensitive to freeze thaws. So we wouldn't want to freeze thaw our samples more than once. So when would you want to use manual library normalization? So you would consider manual normalization if you wanted greater control over how much library you're loading and what cluster density you're getting after sequencing. So for example, if this is the first time loading a certain or preparing libraries from a certain sample type, if you've routinely prepared from genomic DNA, but it's your first time preparing from an amplicon, you may want to know exactly how many picomoles you're loading onto your instrument to get what cluster density. So in that case, you may want to use manual library normalization. And also, bead normalization, again, doesn't work very well if you don't have at least 15 nanomolar library concentration, and also if you don't have a high-speed shaker, or if you're planning to store your libraries for a long period of time, or have to freeze thaw more than once. So in that case, you would want to consider manual normalization over bead-based normalization. So how do you perform manual library normalization? So we need two values to be able to get the nanomolar concentration. So first of all, we would need the average library size, and you would get this by using a bioanalyzer or similar equipment. And then once you have the average library size, you would also need an accurate concentration measurement of our final library, and you can get this value by using a double-stranded fluorescence-based method, such as qubit or picogreen. So once you have these two values, you can use this formula that we're showing you here, and then this will convert the nanogram per microliter concentration to nanomolar concentration. Once you know the nanomolar concentration of each library, you would dilute the libraries to have the same nanomolar concentration and then pool at equal volumes. And then from here, you would proceed to each system's denature and dilute libraries guide to proceed to sequencing on your different instruments. So how do I get optimal cluster density? If you've used bead-based normalization, we have a recommendation of a good starting point for sequencing on the MiSeq. So for Nextera XT bead normalized libraries, 24 microliters of the library pool is a good place to start if we're sequencing on the MiSeq. Depending on the library, it may need adjustments, different dilutions to get optimal cluster densities, but 24 microliters is a great place to start. For the different sequencers, we would be following the protocol B in each system's denature dilute libraries guide, and then there are ranges that are provided in each guide. If we use manual normalization, again, depending on the average fragment size, we have different concentrations that we recommend for optimal cluster density. So one thing to consider is that if we have short library fragments, they will cluster more efficiently on our various different platforms, whereas longer library fragments will not cluster as efficiently. So depending on the average library size, you may want to load a different loading concentration. So you can look at table 1 here, and you can see that for the different library sizes, we have different concentration ranges that we recommend. You can find this table in the tech note that I linked at the first bullet point there. Another way to gauge how much library you may want to load on your different platforms is to compare your average library size to phi-X. So phi-X is a loading control that we use for our validation runs for the different sequencing platforms, and we have recommended loading concentrations for phi-X on our various different platforms. And phi-X is about 500 base pairs in length. So if our library was shorter than phi-X, then we might want to load less. And if our average fragment size is longer than phi-X, we may want to add a little bit more to get optimal cluster density. So once we've performed our sequencing run, what should our data look like? So we have a lot of great public data sets on Bead-Based Sequence Hub, so please check it out. It's absolutely free. So go to Bead-Based Sequence Hub, and the public data tab is on the far right upper hand corner. And you can go to the public data tab, search for Nextera XT, and you can find example data for Nextera XT libraries sequenced on our various platforms, as well as starting from various different starting materials. So we went over the Nextera XT library preparation workflow. The Nextera DNA library preparation, as we compared at the very beginning of this talk, has a very similar workflow, but there are a couple steps that are a little different. So as we mentioned earlier, the Nextera XT library preparation is optimized for small genomes or amplicaons, and we start with 1 nanogram of input material, whereas the Nextera DNA library preparation, we recommend it for large genomes, and we started with 50 nanograms of input material. The Tagmentation reactions are slightly different. The volumes are, we set it up in a larger volume for the Nextera DNA library preparation. And instead of the neutralization step that we did for Nextera XT, we would terminate the Tagmentation reaction using a CDNA Clean Up in the Nextera DNA library preparation. The PCR cycles are slightly different as well. The annealing temperatures as well as number of cycles are different for the Nextera DNA library preparation. And then the Nextera DNA library preparation, because it normally starts with large genome genomic DNA for the most part, we only recommend a 0.6x AMPure XP bead ratio for this kit. For Nextera DNA, the bead-based normalization reagents are all included, whereas for the Nextera DNA library preparation, it does not come with bead-based normalization reagents, so you will have to perform manual normalization for Nextera DNA. And so with that, here are some additional resources you would want to check out. Go to the first link here to check out our expert video tips. And then we have a lot of great tech notes and other useful documents that I did point out during the presentation as well. Again, like I want to stress, take a look at the Nextera XT Library Prep Tips and Troubleshooting Guide. There's a lot of great information here, how to get an optimal library. And then we have a new tech note that came out recently for comparing standard or manual and bead-based normalization on the Nextera XT library preparation, so please take a look at that one as well. And then we have some support bulletins here that are useful for the Nextera XT library preparation, but the bulletins are constantly updated and revamped, so always constantly go and check if there are new bulletins that you might find useful. So with that, you should now be familiar with the kits that are required to complete the Nextera XT sample preparation, the steps of the sample preparation process, the best practices associated with each step, and then when you would want to use Nextera XT versus Nextera DNA. All right, and with that, I'd be happy to take any questions.